Hello, everyone, and welcome. Today, we're going to talk about the innate immune system, and with that, the complement system. Now, again, the past lecture I realized could be a little bit overwhelming, but I encourage you perhaps to look at it after you've become a little more comfortable with the things we talk about over the next couple lectures. That was just to kind of frame the entire immune system in very general terms, and now we start to go into a little bit more specifics, right? So the innate immune system is a bit much, so we divide it into two lectures, and that's okay. So let's begin. So the innate immune system is the older of our two immune systems. We have an adaptive immune system. We have our innate immune system. Innate immune system is very innate, and that it's people who have innate immune system within the same species is pretty similar individual to individual, and many organisms have innate immune system to some capacity. Meanwhile, adaptive immune system is mainly among vertebrae, or vertebrates rather, with the vertebrae, right? Um, and adaptive is, like the name says, quite adaptive. So there's a lot of variability and specificity, right? As opposed to innate immune system is pretty much fixed. And what does that mean to be variable or fixed as an immune system? What does that mean? What are these abstract terms? This refers to the nature of the function of the cells and the proteins. They can pretty much send out like a blanket cover attack that pretty much like a one size fits all type mindset as far as system mindsets go, right? So innate immune system includes the complement system, which is a group of serum proteins that poke holes in pathogen cell membranes, professional phagocytes, and so these are phagocytic cells who perform phagocytosis, mean they eat things, right? And then natural killer cells. Now, the innate immune system does not include B cells and T cells. They're part of the adaptive immune system. So it's very important to be able to differentiate these two systems and be able to say whether or not something is innate or adaptive. And then as we get very specific, I don't think it's going to be this lecture, but you're going to see some cells, they work at the interplay of the two systems because the two systems need to be bridged, right? So that's what that is. So the first thing we're going to talk about in the innate immune system is the complement system, the first thing up here in the first bullet point. Complement is a big mess of letters and numbers. So I encourage you to just repeat, 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 repeat until you get it. And then eventually it seems like procedural desk work. Very routine. But in the beginning, if you're a little bit intimidated, don't worry. I think most people are. Even the people who've memorized it, I think, can get intimidated still just by all those letters and numbers. Again, the way immunology, the things are named is just typically like the lab names, and there's really not that much education-friendly names at this point in the field's development. It's a relatively recent, in some regards, we can say that. I don't want to get into the history. So the complement system is a group of serum proteins that poke holes in pathogen membrane. However, there's different subtypes of complement pathways, right? And so there's three. There's 20 serum proteins. We're not going to talk about all of them, but we're going to talk about the one that pertain to the three main pathway that we're talking about. It's going to be the alternative pathway, the classical pathway, and the Mannose binding lectin pathway. If I confused you, what does oldest and discovered first mean? Alternative means that evolutionarily, alternative pathway is the oldest evolutionary pathway, as opposed to the classical pathway. It was discovered first, but it wasn't like the first evolutionary. Hence, these names don't always correspond. And you also have the Mannose binding lectin pathway. So please don't get overwhelmed. I'm just going to warn you in advance. It's going to be a lot of letters and numbers, but we will walk through it. I promise you. So the alternative pathway is where we will start. And again, recall, alternative pathway is the oldest evolutionary pathway. For this reason, I've decided to choose it first. So at any given moment, including right now, your plasma is full of a protein called C3. And that protein is spontaneously breaking apart every single moment, like always, like always, like every minute of every day, of every second, of every hour, of everything, right? So C3 is always breaking apart in your serum, always, just constantly. And it's breaking apart into two fragments, C3A and C3B, right? C3B is sticky. It sticks to hydroxyl and amino groups found on bacterial cell membranes. if there is a bacterial cell membrane because of the bacteria inside of your body, some of this spontaneously breaking down C3b will stick to that hydroxyl or amino group found on the bacterial membrane surface. I don't think it's too important to know if it's hydroxyl amino group. Just know it's bacterial cell surface. So C3b will bind to the bacterial cell surface. If not, C3b just breaks. If it doesn't bind the bacterial cell surface, C3b just gets destroyed. No use, and then more C3 will come in and break out, and it's always replenishing, right? But it's very key. We speak, it's always happening. C3b only has 60 microseconds to bind bacteria. Otherwise, it binds water and gets neutralized. So that means that's why it's important that C3 is always spontaneously breaking apart, because there's literally almost close to no time for C3b to hit what it needs to. But as you can imagine, because it's always ongoing and always breaking up, just by proximity and whatnot, it is quite useful. So C3b binds another protein called B. I know, just letters and numbers. So now you have a complex on the bacterial cell surface called C3b, okay? Or C3b, right? So you just tack on more letters. That's why the complement looks funny, looks to learn about. So then you have something called factor D, right? I use the mnemonic D for dice or destroy because factor D is a very dicing, like cutting up the onions very small, finely. Does just that. Factor D cuts up B, right, into different subcomponents, and of that we talk about B of B, right? Therefore, you get C3BBB, okay, because B was snipped off via the dicey factor D to get the B component. So now you have something called C3BBB on the cell surface of the bacteria. C3BBB is called a C3 convertase. It's very important that you know that this C3BBB is the C3 convertase of the alternative pathway. Not all pathways have the same C3 convertase. What does C3 convertase even mean? It is an enzyme that can enzymatically break down C3. Now you can be like, oh, but Khaled, I thought C3 is breaking down spontaneously. Why do you need an enzyme to break it down? Yes, you can say that's very valid, but this enzyme is very efficient. It will break down C3 even more efficiently than the spontaneous breakdown. So therefore, when there is confirmation that there is a bacterial cell surface because C3b did bind to something initially, and this complex forms, the presence of this complex is a testimony to the fact that there is a bacteria in the system. Therefore, the innate immune system wants to go all out, bring out all the C3s and to break them down, right? That's kind of like the rationale behind there. A bit dramatic, but it's important. It's self-amplification, really. So C3BBB as a C3 convertase will break down even more C3 into more C3b and more C3b, which will make more complexes by binding B, then cleavage by factor D and everything, okay? If you could understand this part, the rest of complement is cake. So again, C3BBB, his C3 convertase, splits up C3 into C3A, C3B, right? It's more efficient than spontaneous breakdown. Some of these C3b will plant themselves to form on the bacterial cell surface to form more of the complex C3BBB. Some of that C3b will actually bind to the original complex, and that complex will then have two C3bs. So there's different ways you can say. You can say C3b2bb, or you can say what some books say C3b-BB3b. Because now there's two C3bs on the component. What's the significance of this? This is an upgraded form of the C3 convertase. Instead, now it's a C5 convertase, and C5 is another complement protein. If it's a C5 convertase, that means it can break down C5 into C5a and C5b. Okay. C5b, very similar to C3b, plants itself on the bacterial membrane, except instead of attracting factor B and then factor D and all the, B and then factor D and all the things, C5b follows its own story, and the story is very easy. So B is for build. That's the way I tend to remember it. There is an exception later on, but I don't want to talk about it now. So usually the B fragment is the part that builds the complex on the surface. C5b builds itself into the membrane. Okay. So if we can get that straight. So let's just say we have a bacterial membrane here. Okay. And then you get this protein C5b. Let's just say this is C5b. C5b will recruit its friends. C6, C7, C8, and C9. C9 though is interesting because C9 is like a stake, like the one you like use to kill vampires. Its prongs inside of the membrane. Okay, it sticks itself, and then the C9 is actually going to polymerize. It's going to form copies of itself. So you can really imagine it. It's going to form like a type of channel, like a pore. So it's just like a side view, and then something can go through like there, or rather, because it's going to pour out of the membrane, it's going to go like that, right? Or if you look at it from like top down, it looks like a flower almost, right? And then like that white in the middle is the hole, right here. So it's a pore. That's literally what it is, right? So C3. C5b recruits C6, C7, C8, and C9. C9 is going to polymerize and form a pore. This pore is called the membrane attack complex. And this is very important. You know what it is, because this is the whole point of complement. Mac and cheese, MAC. I don't know, however you want to remember it. And so because there's now a pore in the pathogen membrane, the innards gush out and the pathogen dies. And that's literally the whole point of complement. The whole point of complement is to form membrane attack complexes. Okay? Very good. So now, how do we clear all drawing? Okay. So just again to recap, C3b2BB is the C5 convertase, breaks down C5 into C5A and B. C5b is the one that builds, it plants itself on the membrane, recruits C6, 7, 8, 9, 9 polymerizes, forms a pore, membrane attack complex. Good. That's the alternative pathway. Now, we have this curious thing. If C3 is always spontaneously breaking down, what's like the guarantee that, let's just say we do have like an amino or hydroxyl group on our own cells or something of that nature, what stops complement, who's always active, from making pores in our own cells? Valid question. There are 3 well-known mechanisms in which the body ensures complement does not make pores in our own cells. One is called MCP, membrane cofactor protein. Our self cells have this protein littered all over them, right? And so just like complement is all over the serum, this protein is all over our surface of our cells. And basically, if C3b falls onto our cells, this MCP protein clears it up, just to make sure that there is no C3b implanting and possibly starting a chain of events that lead to membrane attack complexes forming. There's something else called decay accelerating factor, this attacks C3 convertase itself. If C3 convertase is like present on a self cell, or just, you know, this DAF will essentially render the C3 convertase useless. And if there's less C3 convertase, less C3 is being broken down. So it also acts as a break on the feedback loop. We also have something called protectin, which is the CD59 is the other name. So see, this is one of those things where actually to give it a standardized name, easier to remember, but sometimes they just keep the CD name. CD59. It prevents C9 from bonding to C5, C6, C7, C8 complex. And remember, C9 is the one that polymerizes, therefore, if no C9, there's no MAC. Then the mnemonic I like to use for that one is CD59 has a 9, which blocks off C9. So this is the key ideas. You can read over them. But this is the key ideas of the alternative pathway. Alternative pathway is always on because C3 is always spontaneously breaking down. Okay. So now we have another pathway called Mannose binding lectin pathway. So again, because it is a complement system, the end goal is to make a membrane attack complex. However, the means in which it does it is different. And so this means here is actually rather clever. And I'll tell you why. Mannose binding lectin, MBL, is produced in the liver. It's a lectin, and lectin is just a protein that can bind the carb, like a sugar, right? And this lectin, what it binds is mannose. And mannose is a sugar found on pathogen cell surfaces usually. A lot of human cells typically do not have mannose sugars tethered to them, but bacterial um, tend to, right? So therefore, just because the nature of mannose binding lectin being specific to something usually found on bacteria, evolutionarily adapted, uh, MBL is very focused. It's not always like spontaneous like C3b. No, no. If it finds the mannose binding lectin can bind it, and it already knows that this is a bacteria because you don't expect your human cells to have mannose, right? So MBL is going to associate with two proteins called MASP1 and MASP2. Please, just I don't want to confuse anyone, but last lecture we spoke about RAG1 and RAG2. Those are somatic hypermutation. Somatic recombination enzymes. These have nothing to do with these. I know just because they sound kind of similar and have one and two, but they're not the same. Don't confuse. Two proteins called MASP1 and MASP2 associate with MBL. MASP2 specifically is proteolytic, so I think of them like wasps, like wasps, like they fly in and they ambush MBL and they grab onto it. Then MASP2 is like this, has like like the stinger, so it's like sharp and like cut stuff up, or just go with it. And so MASP2 bound to MBL becomes proteolytic through conformational changes. So proteolytic means it can cut things up, right? So it can begin the complement cleavage routine. Now there is some differences between different books on what they say that what is it that MASP2 cuts up. Some say it is C3 into C3A and C3B, and it's just like what we saw before with spontaneous C3 breakdown or C3 convertase. However, the one I tend to believe and the one that I feel like from other books, and perhaps just know for your own viewing pleasure, or depending on how specific you should know it, that MASP actually acts on a different complement protein, and it's called C4. And it's going to cleave, MASP2 is going to cleave C4 into C4A and C4B, and the schema is very different. C4, instead of binding factor B, it's actually going to bind something called C2. And so C4BC2 is going to form. And instead of factor D, actually MASP acts again, and it's going to attack the factor 2. And so before, I usually told you that the B component is what builds the complex. C2 is the only exception where the A component builds the complex. So actually in MBL and also classical, you're going to see that it's different in this regard because you have C4B, C2A. And C4B, C2A is a C3 convertase of the mannose binding lectin pathway. Meanwhile, remember for the alternative pathway, it was C3B, BB. And then to make C5, C3, B2, BB. Right. So now you have this C3 convertase, and it just does the same thing as any other C3 convertase. Breaks up more C3, C3A, C3B. Some is going to deposit, the C3B is going to deposit on the membrane, recruit, C6, 7, 8, and then 9 polymerizes, forms a pore, right? So really the end goal is just to form the membrane attack complex by getting that C5b to deposit and then to recruit all its friends and then C5 to polymerize. There's just different ways of breaking down C3. There's spontaneously, and then you have this very targeted method for organisms that have mannose on their cell membranes. So these are some pathogens that contain mannose on their surface. Some are fungi like Candida that causes like thrush. Some like thing you have to use nystatin for. Not that I use it, just if someone has to use it. HIV, it's encapsulated type RNA virus. Influenza, Salmonella is bacteria. It's one of those gram negatives. Streptococcus as well. But Streptococcus on the other hand is a gram positive cocci, and then you also have parasites like a multicellular organism. These are just different things, so it doesn't have to be bacteria. It can be any. A lot of pathogens have mannose on their cell surface. So now we end on the classical pathway. It's very similar to the MBL pathway, and evolutionarily they're quite simple. But instead of MBL doing the work on the MASP proteins, it's actually going to talk about the antibodies, which I was talking about earlier. Remember, one of the second function of the antibody was to scaffold complement proteins, right? That's exactly what we have going on. So C1, like we're talking about C2, C3, C4, C5, all these. Where's C1? Well, C1's on the classical pathway, if that helps, right? C1 will bind to the pathogen surface. However, in order to bind to the pathogen surface, it needs a scaffold, and so the scaffold will either be one IgM, which is a pentamer, 10 binding sites, or two IgG molecules, so 4 binding sites total. So the key thing to know here that one IgG molecule is not going to cut it because that's only 2 binding sites. You technically need at least 4 from the two IgGs. So because each has 2 binding sites, when you have 2 IgGs, it's 4 binding sites, or just one IgM, which is 10 binding sites. So you need immunoglobulin scaffold for C1 to bind. If C1 can bind its scaffold, then the C1 will, do its purpose, and then the C1 acts just like C1 bound to an antibody acts just like MBL bound to MASP2. C1 will cleave C4, C4A, and C4B. C4B will go on to bind C2. Instead of, again, MASP, it's C1 doing the job. C1 is going to come in, or rather C1Q is going to come in and bind or cleave the C2 on C4BC2 to get C4BC2A. That's C3 convertase, splices C3A into C3B, C3 into C3 and C3B. C3B is going to deposit on the membrane, and then recruit 6, 7, 8, 9. 9 polymerizes, forms a pore, right? So it's a bit like I said, I think it's better to learn MBL first, and then just for classical, just know that instead of Mannose binding lectin and MASP, it's going to be a C1 with this proteolytic C1Q domain specifically bound to 2 IgGs or 1 IgM. Okay. Sorry if I confused you. I recommend you just go through the thing a couple times, and then you'll get it. I guess, again, once you get to that part, when you have a C3 convertase, no matter what it is, whether it's C4B, C2, or C3B, BB, either case, once you have a C3 convertase and you get C3 into C3A, C3B in an amplified form, and C3B can deposit and then form the C5 convertase, and you have C5B on the membrane, recruiting C6, 7, 8, 9, and whatnot, and then the polymerize. As long as you have that happening, the steps are generally the same after that point. Before that, you have some variety. And like I said, classical and MBL are very similar. Again, it's mouthful, so I'm sorry if I confused you, but I hope if me casually talking it out maybe helps a little bit. I'm not a genius. I'm actually quite the opposite. But if you guys do the thing enough times, one of those things like you just kind of stays. I won't say like riding a bike, because in my childhood I actually had difficulty learning how to ride a bike, but now it's quite enjoyable. So again, this is just a review from the other thing, or actually no, not really. Part of it is review. What does complement do? Aside from forming the membrane attack complex, the complement proteins actually have some other roles. C3b is an opsonin, right? So just like some antibodies can act as opsonins from the first lecture, C3b can also act as an opsonin when it's rendered into its IC3b form. And so this actually makes it easier for phagocytes to take up cells that have IC3b deposited on them. Okay, so it's definitely good to know C3b, or specifically IC3b, is an opsonin. Also, we always talked about the B fragments. We didn't really talk about the A fragments, except C2A, which is part of the C3 convertase and MBL and classical. But aside from this, what happened to C3A and C5A? These are actually A, and A for anaphytoxin, B for build is the mnemonic I use, but they're anaphytoxin. So basically, they act as chemical that can signal other white blood cells to come to the site where a complement is being activated, because chances are where complement is being activated is the site of a bacterial or viral or parasitic infection, right? So that's what we call chemoattractants or chemoattractants. Right? Too much C3A and C5A can actually lead to anaphylactic shock. Hence, they have the name anaphytoxin.